The History of Gel Electrophoresis by Zoe, Winnie, Madeline, and Melissa.
Gel electrophoresis is a procedure used in molecular biology to separate and identify macromolecules by size and charge. Examples of macromolecules include DNA, RNA, and protein. The word electrophoresis comes from the prefix electro and the suffix phoresis. Electro means something that is related to electricity, and phoresis means migration or being carried. Together, the word electrophoresis means the movement of charged particles in a fluid or gel under the influence of an electric field.
We've covered what electrophoresis is and what it means, but what about the gel that's also involved in this process? The gel matrix is a porous substance that serves as a molecular sieve, just like the picture on the top right corner. Because of this, the gel is able to provide resistance to the migration of the molecules. Moving on to the process of gel electrophoresis. First, we need to know that the nucleic acids in which are used in gel electrophoresis are negatively charged. The overall charge of the molecule is proportional to its mass, which creates a ratio of charge to mass. As a result, the mobility of the nucleic acid in gel electrophoresis is determined mainly on its size. When the electric field is turned on, the nucleic acid begins to migrate from cathode to anode, which are the negative and positive electrodes. As you can see on the picture on the right, the smaller pieces of the nucleic acid, which are the blue fragments, are closest to the anode because it moves the fastest.
Now here's an example of gel electrophoresis at work. Here, instead of using DNA, we are putting food coloring in the gel. When we turn the electricity on, the colors dispersed, since each color moves a certain distance. For example, in this video, yellow and red go quite far, while blue stays close to the beginning. Now let's dive into the historical background of electrophoresis. Yay! Ferdinand Frederick Roos, a Russian chemist, started the discovery of electrophoresis about 200 years ago when he studied the movement of colloidal clay particles on an electric field. A colloid is a mixture in which one substance of microscopically dispersed insoluble particles is suspended throughout another substance. From his observation, he realized that the clay flowed opposite to the flow of water. This led to the discovery of electroosmosis. As you can see from the picture, the applied electric field flows opposite to the flow of water. Protein was first separated in 1930 by Arne Tiselius, a Swedish biochemist. In his experiment, he took human serum and used electrophoresis to separate it into 5 proteins. Because of this, he eventually won a Nobel Prize in 1948. During the time of World War II, scientists managed to find a way to perform electrophoresis with paper. With charged particles, they called it paper electrophoresis. With uncharged particles, it was called paper chromatography. A few years later, the scientists replaced paper with nitrocellulose membranes, which are commonly used to immobilize nucleic acids. Oliver Smithies, a British-American physical biochemist, made many important discoveries in 1955. He first realized that proteins can be separated on gels, or more specifically, starch gels. He then proved that separation with electrophoresis is molecular size dependent. He also managed to separate serum protein, but on a 2D spectrum, by first separating it on paper and then starch gel. These discoveries led to him receiving the Nobel Prize in 2007. Unfortunately, Oliver Smithies passed away last year at the age of 91. Also in 1955, a scientist named O. Farrell used synthetic gels that were synthesized from a polymer called polyacrylamide. This was a 2D process that used the isoelectric focusing of proteins, a technique used to separate different molecules by isoelectric point. An isoelectric point is a pH at which a particular molecule carries no net electrical charge. In the 1960s, Archer Martin used agar gels as an electrophoretic medium. Currently, we are still using these gels for the separation of DNA. Agar gels are a jelly-like substance obtained from algae. With agar gels, in 1964, scientists managed to separate RNA successfully. In 1981, they invented capillary electrophoresis, which is a separation method performed in capillaries, or in micro and nanofluidic channels.
The role of CE in the Human Genome Project has been covered by Jung and Devaki. The Human Genome Project finds DNA sequences associated with diseases such as diabetes and cancer. It also provides important new technologies for the private sector, including agriculture and medicine. The original 16 capillary systems have been replaced by 96 capillary systems to improve faster sequencing. The Agilent bioanalyzer, when launched in 1999, was the first commercially available instrument to use chip technology for the analysis of DNA and RNA, as well as being able to perform SDS-Page electrophoresis on a benchtop system.
That's it for now, at least until the technique of electrophoresis improves and evolves. Thank you for listening.